Hello, everyone. Welcome to Life Science Across the Globe. I'm Janine Stevens, one of the Genelia organizers of this series. And on behalf of all 8 sister institutes, I welcome you to today's event hosted by EMBL on learning from the planet to heal the planet, microbial ecosystems. As always, I encourage you to visit lifescienceacrosstheglobe.org to see our full lineup of monthly events for the year, to subscribe to the event calendar, and to view recordings of all of our past events. We would also love your input on the brief survey that we'll post in the chat box towards the end of today's session. As a reminder to trainees, you are invited to stay on the call after the event today for a special Meet the Panelists session. Also, to all of our audience, please go ahead and write your questions in the Q&A box at any time, and our moderator will ask them during the Q&A period. And now I want to turn it over to Cornelius Gross, interim head of EMBL Rome, for some opening remarks. Cornelius.
Hi, thanks, Janine. Welcome on behalf of the European Molecular Biology Laboratory to this Life Science Across the Globe event. This is the second edition, actually, of this Learning from the Planet to Heal the Planet series, which our organization has been hosting within the Life Science Across the Globe series. In September of last year, we hosted a similar event. It was focused on marine ecosystems. So if you're interested in that, I urge you to check that out. It's online. And today, the topic will be the fascinating field and world of microbial ecosystems. So the series is a part of a growing movement in the life sciences that's trying to link molecular biology work, which has traditionally often happened in the laboratory, with field work to really kind of understand the molecular mechanisms of how organisms interact in their natural context, how they interact with each other, how they interact with their physical environment. And we have a really exciting lineup of speakers today. And it's a special pleasure of mine to introduce the moderator of the event, who is Ron Milo. Just to tell you a few words about Ron. So Ron was trained as a mathematician and a physicist. He completed his master's degree, which he himself said was both extremely frustrating and also mediocre. And then he did manage to move on. He eventually became a faculty member at the Weizmann Institute in Israel. And there his lab is interested in using synthetic and computational biology approaches to address challenges in sustainability. He's most proud of work in which he, in his laboratory, were able to make E. coli into an autotroph. That means that they're able to survive just on sunlight. But for all of those of you out there frustrated with the publication process, he wants to reassure you that that publication actually was rejected by the major journals, Nature and Science. So thanks, Ron, for being with us today.
Thank you, Cornelius, for the introduction. Thanks, everybody in the audience for joining us together with the 3 distinguished speakers. Before we jump in, so we'll be hearing today about environmental challenges and how science and microbial research in particular could really help. But I think before we plunge into all the environmental challenges, which are huge, I think we should first appreciate how lucky we are. We are not only blessed with a fantastic planet, but we also live in a time where most of humanity enjoys food, water, the ability to heat and cool their houses, as well as, mobility, communication like we're doing now, and education like never before. What we enjoy comes with a toll of a global environmental crisis, which is one excellent reason to have this session that we'll be having today. And so I think we should begin the meeting with a sense of how blessed we are, and also with a sense of urgency, because a lot needs to happen in the next 10 years. And it's crucial that science will play a key role in that. And within science, I think the over 100 people that joined us now are the people that I hope will be joining the effort. And perfectly suited to begin this journey is Professor Dianne Newman from Caltech. She's one of the world's leaders in how microbes shape our biosphere. She also shared with me that she was a humanities major in college and never would have dreamed she'd wind up as a molecular geobiologist, as she thought biology and geology were boring and all about memorization. By chance, she discovered microbial metabolism and genetics in graduate schools, and finally saw the light. So Dianne, please share some of the light with us. Thank you.
All right. Well, thank you. Thank you all for coming to this session. I loved your introduction, Ron. I think it set just the right tone. And it's really my honor to begin by telling you about the impact and the promise of microbial metabolism. So to get some perspective, let's start with the view of our planet that Neil Armstrong had when he landed on the moon. And something I think important to appreciate about this picture is that Neil Armstrong is in a spacesuit because for his metabolism, he cannot survive without oxygen. And in fact, oxygen is so intuitively part of how we think about life that when we look for signatures of life on exoplanets, we often look for, in the infrared planetary spectra, signatures of ozone, for instance, like you see here in Earth. Of course, water is really important for habitability as we know it. And when we see signs of ozone, this implies that there's oxygen in the atmosphere, and we think, well, maybe that planet has life. And yet what I want you to appreciate is that had we been looking for oxygen on Earth, we would have missed the first billion years of life's history. And so let's just take in the timeline of Earth history and recognize that the Earth is 4.5 billion years old. Microbial life is thought to have originated around 3.8 billion years ago, but it wasn't until roughly 2.5 billion years ago that we see signatures of oxygen in the atmosphere. Now, that means there was at least 1 billion years of the history of microbial life on the planet where they were subsisting through metabolisms that trafficked in things other than oxygen. And though animals evolved on a microbial world after oxygen came on the scene, as we know from some of the remarkable progress in studying the human microbiome, we've got a ton of microbes within us, and there are even many, many more within the microbiome of the Earth. And within both of these habitats, both within the human gut and within many locales on Earth, there remain many anaerobic habitats. So there's all sorts of remarkable ways that microbes have figured out how to conserve energy in the absence of oxygen. Now, this is important because through this time, microbes have been very clever. And these different strategies that they've evolved for energy conservation have led and continue to lead to profound impacts for the planet, as I'll give you a few examples of in just a moment. But what I want to also impress upon you is the fact that these diverse microbial metabolisms have incredible potential to be harnessed for a variety of purposes to help the planet going forward. So let's think very broadly about metabolism and energy conservation, just to put this into context. So for us, our mitochondria are utilizing electrons ultimately derived from organic carbon and oxidizing this electron donor with oxygen. That's the electron acceptor, converting the substrates into CO2 and water, which chloroplasts found in plants recycle. And so this is the familiar cycle between animal and plant metabolism that many of you are familiar with. Of course, I want to impress upon you that the mitochondria and chloroplast are nothing more than ancient bacteria that were engulfed once upon a time in an endosymbiotic event. And what's critical to realize is that the substrates used by diverse microbes are numerous, and it's really all about the ability for redox chemistry to be captured in a way that promotes energy conservation. So redox chemistry is the reduction, the gaining of an electron, coupled to the oxidation, the loss of an electron. And so here, written generically, I have the electron donor as A reductant and the electron acceptor as B oxidant. Now, a remarkable diversity of substrates can serve as electron donors or acceptors for microbial metabolism, ranging from organic solvents to minerals to gases to even toxic metalloids. And the particular way in which energy is conserved from these various substrates varies depending upon the substrate, but for metabolisms that involve energy conservation at the level of the membrane, they all work something like this, where the reductant, the electron donor, interacts with an enzyme in the membrane, passing electrons through this enzyme coupled to ion translocation. And typically this is either a proton or a sodium ion outside of the membrane. And then the electron is passed through carriers down the chain, ultimately to the terminal electron acceptor, or B oxidant, which gets reduced. And in this process, what winds up happening is a wonderful potential is generated around the membrane like a battery that can be harnessed to drive the work of making ATP through ATP synthases embedded in the membrane. And this leads to energy conservation. Now, the effort to understand how in molecular detail this works is absolutely fascinating, and it provides rich opportunities for impactful discoveries and application, as you're going to hear later in this seminar. So let me give you just a few examples of big picture effects from different electron donors and acceptors. So as I said at the beginning, our oxygen in the atmosphere is a biosignature. Specifically, it is the remnants of the invention of oxygenic photosynthesis, where water was used as an electron donor to support photosynthetic growth. Yet, prior to this particular form of photosynthesis, there were others, including some that utilized ferrous iron as an electron donor in what is called anoxygenic photosynthesis. And there's good reason to believe that going way back in time, over 3 billion years ago, some of the ore deposits on Earth, such as this here, a banded iron formation from Western Australia, may indeed have been deposited due to this alternative form of photosynthesis. Now today, we know that on short time scales, microbes can actually remediate waste dumps, such as in the case of the Deepwater Horizon oil spill in the Gulf of Mexico, where petroleum toxic compounds like benzene and toluene have been degraded because they're used as an electron donor, supporting the growth of a diversity of microbes in this environment. And yet microbial metabolism doesn't always lead to good things. What's also true is that in certain environments, such as in groundwaters of Bangladesh, the input of organic carbon exceeds the amount of oxygen present in these systems, so that alternative electron acceptors like iron minerals or even the toxic metalloid arsenate begin to be used to oxidize those organic substrates. And this can tragically lead to the mobilization of toxins. Now, understanding how these things happen can allow us to invent diagnostics to know when certain activities are occurring and even think forward to how we might control them. And what I want to pose to you as a challenge to the young people in the audience is what I consider an important and unresolved question in microbial ecology about the future, involving global warming potential and microbial feedbacks. But first, let's step into the past to appreciate this metabolism and go to Lake Como in northern Italy in 1776, the same year as the Declaration of Independence in the United States. Here we have a picture of Alessandro Volta poking at the sediments in this lake, and what he's doing is collecting in a glass jar bubbles from these sediments. Through ingenious experiments that he did, he was able to figure out that the bubbles contained the gas methane. And this was the discovery of methanogenesis, an incredibly important metabolism catalyzed by microbial archaea. Here you see an example of a Voltaflame experiment being performed today. So I hope you heard the dramatic sound effects coupled to that. This is an incredibly fun thing to do. Be careful, don't singe your hair if you do it. But what it shows you is that there's an incredible amount of methane that you can even find in simple bogs or lakes in freshwater environments all over the planet. While it's dramatic and while it's fun, it also connects to something actually rather serious, which is that we now know that the 20-year global warming potential of methane exceeds by 80 times that of CO2. And why do we care about this? Well, the reason we care is that if we look in the northern hemisphere at the frozen permafrost, whether it's in the marine or in the terrestrial realm, what we have is an unknown, which is that this permafrost is thawing, and within these permafrosts are a lot of tons of carbon. So this has been quantified nicely in this study by Crowther et al., where what you're looking at is the global carbon stocks as a function of latitude. And I want to focus you on this rightmost portion, which is where you see by far the most carbon stocks being present underground here in these northern permafrost latitudes. So what will happen as they warm? We can even see evidence of warming today. Here's a photo of slumping permafrost in northern Canada. Will CO2 be released? Will methane? Or will microbial communities adapt so that there's a steady state that's maintained? We don't really know, but we need to know. We want to understand how fast changes might occur here. And importantly, we need to be able to understand what might limit them. These are very critical questions for the future that I hope some of you might begin to get interested in following this presentation. Now, for any question such as this, to be able to predict how microbes will affect the planet and to control and harness their metabolism, we need to do the following. First and foremost, we must understand in mechanistic detail how they catalyze reactions of interest. What are the enzymes? What are the cofactors? What are the rates of these enzymes? What limits them? How widely distributed are they? These are questions that our following speakers, Rob and Toby, are going to address in their talks. In addition, we need to understand the regulation of these processes, and we need to think very carefully about the potential for quantitative impact, something our host, Ron Milo, has really led us and inspired us to do as a field. So for the remaining time, I want to give you just a quick taste of research from my lab where we were captivated by questions involving soil bacteria, and particularly those that produce colorful pigments and ones which change color over time. Here you see a lovely plate of the Munch's cream rendered in soil bacteria making pigments. This was done by Tomislav Ivankovic, a scientist at the University of Zagreb. And the type of pigments that interest me in my lab are ones such as this that you see here in the middle, which changes its color over the distribution and length of this colony. An organism that we study in my lab is Pseudomonas aeruginosa, and we play games with it where we can introduce oxygen by aerating it, and it turns color. And then as it sits, the color changes and it goes back to where it started. Now what's happening here is that this organism is producing a redox-active pigment called phenazine, which when it is produced in its reduced state it is clear, but when oxygen is introduced into the system, it oxidizes the molecule, turning it blue, and oxygen in turn receives those electrons and becomes superoxide. And it's the generation of superoxide that has made people previously think phenazines are antibiotics and toxic. Yet we questioned whether this was the full story. And the reason we questioned this is that it had been known for a long time that the production of phenazines were regulated by quorum sensing, that they turned on as cells were reaching the tail end of growth, using up oxygen. That's when they would make these compounds. And then they would start recycling them. So why does this matter? Well, in the absence of oxygen, having a self-made electron acceptor would be incredibly convenient, but only if one could recycle it. And so that's a challenge mechanistically. How is it at a distance, through a process called extracellular electron transfer, do these beautiful pigments serve as electron acceptors to the cell and become regenerated? Something I'm happy to talk about later. Be the mechanism as it may, what this leads to is the conservation of energy, which enables the sustenance of slow growth in communities that are oxygen limited, such as microbial biofilms, which form on a wide diversity of substrates. Here you see a scanning electron micrograph showing you all of these hundreds of cells in a biofilm. And the important number to recall is that after the first 10 microns, the pace of aerobic respiration outcompetes the diffusion of oxygen into these films. So the guys in the middle are literally struggling for survival. They need an oxidant, and it's under these conditions that they cleverly are able to make and recycle their own. And understanding this, I think, is very interesting, both fundamentally, because slow growth really is the dominant pace of life in the microbial world, yet very poorly understood. And it's also practically important because slow growth often underpins antibiotic tolerance. And so having a mechanistic understanding of what sustains these populations can open up new approaches to controlling biofilms in both nature and disease. Now, thinking about other contexts where phenazine recycling is important, we can replace oxygen with an iron mineral, such as found in the soil. And oftentimes, phosphate is attached to iron minerals, such that when phenazines transfer electrons to these iron oxides, they liberate phosphate due to mineral transformations that make it no longer able to be bound as well. And this potentially is something useful that we can consider harnessing. And so it raises the question, can we engineer organisms to generate these phenazines under environmentally relevant conditions? And might this provide an opportunity for bioaugmentation that would enable greater access to phosphorus, a limiting nutrient in many agricultural contexts? Might it make it easier for us to fertilize fields in a way that is more sustainable and less wasteful? Well, we're just beginning to get into these questions. Through bioinformatic approaches, we've been able to quantify in global metagenomes the proportion of bacteria in rhizospheres. That's the zone of the soil in the vicinity of plant roots. And what we find is that phenazine production is widespread, that roughly 1% of bacteria in rhizospheres encode the machinery to make these molecules. So this gives us hope that this might be something to leverage in this type of context to further the ability to utilize phosphorus more sustainably. Now, a final example I'll give is if we replace that iron mineral with an electrode. Same thing can happen. Phenazines can be reoxidized if the electrode's potential is poised appropriately. Only in this case, the product of the oxidation of the phenazine is the generation of current. And this then opens up all sorts of application for utilization of small molecules like phenazines to stimulate microbial fuel cells, whether these be fuel cells involved in remote power on the ocean floor or fuel cells coupled to wastewater treatment plants. Certain exciting forays into this area are now beginning in Europe and other parts of the globe. There's a lot to do, but it's a potential to actually invest in more creative energy capture in a wide variety of locales. So with this, I'll just wrap up and leave you with a thought, especially the trainees. And that is that sometimes curiosity about something basic, like how it is that bacteria change color, can have surprisingly broad relevance and open up many more questions and challenges. So I hope in your own scientific careers, you'll have the opportunity to pursue your curiosity. We absolutely need new talent to come into this important realm of planetary biology to bring the power of mechanistic molecular analysis to environmental problems. We need institutional support, so we're grateful to the EMBL, HHMI, other institutions, for promoting these efforts. And very importantly, we need quantitative grounding in global organization because these type of problems transcend what any individual institution can achieve. So I'm highlighting for you two outstanding websites that I'll be happy to talk about more in the Q&A that I encourage you to take a look at. The first one, Human Impacts, put together by Ron Milo and my colleague Rob Phillips at Caltech, has some great numbers explaining to us how human activities are impacting the biosphere in a quantitative way. I hope increasingly we'll see information on this site about how microbial interventions have the potential to impact certain sustainability problems. And lastly, I want to do a shout out to the fungi. This is a great website by Tobias in the Netherlands, informing us about their critical roles. Thank you.
Thank you, Dianne. I think this really shows the path we should take and all the creative ways to look at things and find solutions. And you mentioned EMBL and its important role. And I think that's moving directly to our next speaker, Rob Finn, who is a team leader at EMBL-EBI Center. He's a world leader in microbiome dataset sharing, such as the Magnified database he's leading, which enables all of us to analyze, discover, and compare microbiome data. And I think you'll see examples very shortly. Rob started in microbiology, but after frustrations following 48 hours of continuous protein purification, he came to the conclusion that the wet lab was not where his strengths were. Happily, he has many strengths. And so after his PhD, he changed his career path to bioinformatics. And Rob, please tell us about all the interesting things you found in that new world.
Thank you. Brilliant. Okay, so thank you for that introduction. And as Ron mentioned, I work at European Bioinformatics Institute as part of EMBL, and I run a resource called Magnify. And what I'm going to do is talk to you not so much about the science, although there will be some element of science, but also about how we run these resources. So one of EMBL's core missions is not only to do research, but also to deliver science resources to the community. But what I really want to do is start to try and shed some light on how these huge data sets, these metagenomic data sets, can actually help tell us about how we can heal the planet. So your quick 101 on metagenomics, so for those who may be not so familiar, is on this slide. So metagenomics is really the study of the entire microbial community that's living in a particular ecosystem. Now, on this slide, on the left-hand side, I've got the big three that I always refer to in terms of microbiomes that have been particularly studied. So there's been marine microbiomes, soil microbiomes. Dianne's already mentioned a bit about soil microbiomes, and then the human gut microbiome. But since the field started, when we first started looking into those microbiomes, the techniques and sequencing isn't as cheap as it was, as it is now. And so often people could only look at who was there. They would use a marker gene, the so-called metabarcoding, and actually just use that gene to work out who was present within an ecosystem. And typically that marker gene would be something like the ribosomal RNA, the SSU, but there it would be restricted to just looking at the bacterial component. Since then, sequencing has got much cheaper and the interest in the field has increased. And so now with this diminished sequencing cost, we can actually look at the whole community. So we can look at the viral component and the single cell eukaryotic component. And now we tend to look at the whole sum of the DNA, the so-called metagenomic part. That's where you're looking at the total DNA. You can also look at actually what part is metabolically active by looking at the metatranscriptome, although that's generally a bit harder because extracting the RNA is more difficult than extracting the sum of the DNA. But since the field has expanded and we can start having not only who's there, but what those microbes may be doing, this area, the field has diversified in different applications. So there, on the left-hand side, again, there's the flasks that indicates this notion of green chemistry. Can we find enzymes that can carry out jobs that previously chemical synthesis would perform? Can we understand biofuel production that microbes can be involved in? So can we go into silage heaps and understand those microbes? And the oil industry is also very interested in this. We've heard about how microbes can clean up oil spills, but their microbes are very good at actually going into oil fields and polluting oil fields with hydrogen sulfide, so-called oil field souring, and they also form biofilms on the inside of pipes that cause corrosion. All of this leads to a huge different diversity of microbes, the complexity of the communities, and how we can study them. And what I do is produce a resource that tries to capture all of this data that's coming in across the community and actually provide a free-to-use resource that not only captures this data, archives it with the associated metadata, assembles this data where appropriate, and then provides the analysis. And this is a free-to-use service. So what we do is, that the approaches that we use can be computationally very expensive. And what this allows is us to start democratizing science and allowing those who don't have such facilities to actually partake in this. It also allows people to just go to one place to discover data. Now, I'm not going to walk you through the website. The website is at the bottom here and it will also be available later on. But what I wanted to do is, when we talk about these functional analysis pipelines and how we start thinking about putting these together, what we do is we draw on the experience from across EMBL. So there are research groups specifically at EMBL. So I'll point out initially Per Bork and Georg Zeller's groups, where they're producing tools for the analysis of microbiomes. What we can do is we don't need to invent everything in my group. So we take tools that have been produced by them. So there's one example of the tool we've already included is the MOTOL tool, where it's trying to understand the microbes there from their functional profile signature. And this tool in itself has actually been built from collaboration beyond EMBL, so using SIB and ETH Zurich, there've also been contributions there. And what we do at EBI, we have other teams that are experts. So, for example, the Bateman group produces functional annotations of both proteins and non-coding RNAs, and we bring in those components. And when my team combines all of these to make sure these tools are efficient and comprehensively put together. We have these really large pipelines. I don't want to scare you with this slide. What I wanted to just illustrate from these slides is one of the things that we do is formally describe these pipelines and then make them available such that people can take them and reuse them if they want to extend them or change a tool such for their own use. And it's really important that we have this reproducibility so that people can understand, you know, if you get two different results, is that because the community changed, or is it because there was actually an informatics difference? And this is a really important area where we're beginning to find that actually the informatics can cause what we refer to as confounding factors. And what we want is this full provenance of here's the result, and this is how we got it using these databases at this time. For those who are not so maybe interested in that sort of informatics part, what we do is this standardized view. So you can start understanding the data sets, and we make the data sets all look like one another as much as possible. So in this particular example on the left-hand side, it's a data set that's just become available in Magnify, and it's an upper respiratory microbiome, very pertinent to the current pandemic. And so we can see that this was actually come from the US, and then we have lots and lots of data associated with all the different sequencing runs. But if you want to start accessing that data, you don't have to be an expert. So down the bottom, we have these two buttons. And what we're now doing is trying to allow people to go beyond the Magnify analysis, but simply take slices of the data into things like Jupyter notebooks, where they can then process this. And then they can actually share these workflows in these Jupyter notebooks. So we proliferate this idea of reusing data and reusing people's code and expertise such that we can really understand these communities as a whole. So that's a very brief overview of Magnify. Where I wanted to go next is very much where we're moving to as a resource in terms of as the field evolves, and some of the analysis that we do undertake beyond Magnify. So in this particular slide, there's a very simple illustration of what Magnify does. So we start off with typically short sequence DNA reads. And about 10 years ago, we were purely focused on just analyzing those short reads. But as algorithms have improved and we've got greater access compute resources, we started to move to actually assembling those short reads into longer contigs. At each step along the way, though, as we assemble those into contigs, we'll then push that data set back into the public archives, so the European Nucleotide Archive, such that other people can reuse those. And so once we have those longer contigs, we can start understanding whole proteins, or even these broader biosynthetic gene clusters that Dianne mentioned. But then really where we want to go is not only knowing there's a protein, but where has that protein come from? And this is this notion of clustering contigs we think have come from the same organism. We refer to that process as binning. There may be a bin refinement process where we try and identify contaminating contigs, i.e. contigs that shouldn't have been there. And then we have a set of things that we think are representative of genomes. But because we look at this at scale across many data sets, we can have duplications within those. And so we then try and remove those redundant genomes and choose the very best ones to represent a species. And that's what we're starting to make available in Magnify. So we started to make biome-specific genome catalogs. Within Magnify, when I said this is free to use, the data may be already public, and you can ask us to analyze them, or you may have just conducted an experiment, and you can actually upload that data to us in a pre-publication state, and then you can have it analyzed such it remains private. So following that sort of approach, we've applied this to human gut microbiome data sets. Using that, we've applied this to 12,000 of those short-read data sets. We then identified nearly 2,000 species. At the same time, there were 2 other groups doing something similar. And rather than arguing about whose catalogue was better, we actually amalgamated these all together and actually have come up with a fairly comprehensive gut microbiome catalogue of nearly 5,000 species, of which 80% are yet to be cultured. On the right-hand side of this slide, you can see the phylogenetic tree of those species, and coming off the tips of the trees, this green, and those represent those species that are yet to be cultured. And I think this may come up later, and so I won't go into what that exactly means. But what we can understand though is that in the human gut microbiome, although it's one of the most well studied, there's still huge amounts to learn. But through all those assemblies, and Magnify has now over 28,000 assemblies, we have many, many proteins now. We have 2.4 billion non-redundant proteins. So what can we do with these? So here is one specific example. This was done in collaboration with a company. They have, as part of nylon manufacturing, a precursor is the production of adipole nitrile. And as part of that production, there is a waste stream. But you can transform that waste stream with a nitrulase into something useful. But current enzymes were very, very slow. And so we went mining in that data. And what we could also do, so we went mining in the data, we found 4,000 potential targets. But then we could use the environmental data, such as pH and temperature, to optimize the selection. We had 20 candidates. They were tested. 4 had perfect chemistry and 1 had really conversion rates. This has gone into production as a bioremediation enzyme. Another area where we've used the magnified proteins is through collaboration. So the Holfelder group at University of Cambridge, they've been looking and searching for novel plastic degrading enzymes. So in this top left-hand corner, you've got the petases that are well-known degrading enzymes. And then what they did is they were doing functional metagenomic screens, and they came up with this sequence RR1102. But what they could then do in this sequence similarity network is actually map onto magnified protein sequences and come up with this other sequence, this CI15. And on the right-hand side, you can see an indication of how well these are actually degrading petases. So the best one is already shown in the back in black. And then the one that was discovered in Magnify is the next best one that is out there. Since then, Florian has gone through and actually mutated this and actually now has one of the best performing plastic degrading enzymes out there. And this is how we can start harnessing the microbes. This protein data set has come up in all sorts of different shapes and guises going forward. So many of you have no doubt heard about the revolution of protein prediction from AlphaFold. And so on the left-hand side, we see one of these amazing predictions of protein from the AlphaFold database. So AlphaFold, if you don't know what that is, that's an artificial intelligence system produced by DeepMind to actually try and just determine how a protein folds up in 3-dimensional space from sequence alone. But underneath that algorithm, and I'm not trying to lay claims to anything of the amazing work of the AlphaFold algorithm, but underneath that, the magnified protein database sits, and it is used as one of the target databases. So the way you fold this up is you look for covariance in the alignments to say, okay, these 2 amino acids are changing, then and when you see that, they may be close in space, and that's how they do these correlations. And in the paper, it quite clearly said, these metagenomic data sets, they're really useful for actually probing the space that we don't currently know well, and do make a significant difference to the accuracy of the algorithm. So just moving to a last example of how Magnify then engages the community. So we also work within scientific projects, and I'm just going to illustrate the Hollow Food project to you. So this is about looking at both salmon and chicken production. And the example I'm going to pull on is from the salmon production. And this is about how microbes and the host, in this case, the salmon, interact and how they influence that. And what we wanted to do is start understanding these dynamics. Now, as Ron alluded to, I don't go near a wet lab at all. So collaborators are really important here. So in the middle here, you can see them harvesting gut microbiome data. And these salmon are really important. So they're all in the aquaculture system. And we really need to have different ways of generating protein for actually feeding the world's population. And aquaculture, especially in Europe, is a very key area. And so we want to understand, the influence of the microbes and the salmon in these aquaculture systems. They're called recirculating aquaculture systems, or RASes. And one of the key things that we want to do is look to see how the food influenced the salmon production and the microbiome. So one example of the food additives we tested was actually looking at the introduction of seaweed. Now, seaweed, if you have heard anything about human gut microbiome and people encouraging you to eat things like cabbage or kale, it's really good with the fibrous content and actually promotes microbiome health. The other additive we were looking at is blue mussel. Now, most fish are fed on fish, which sounds really counterintuitive, and we really want to break that cycle. And actually, trying to find alternatives to fish meal is really, really important, and is actually one of the industry has mandated that this must now happen. And so what I wanted to do, and this is really hot off the press because I got this last night, is just to show you one graph about how we've been using this blue mussel protein. So in this graph, what we have along the x-axis is at 0, there's no blue mussel, and then we increase more and more blue mussel into the meal. So 0 actually represents our control. And then each one of these diamonds represents the average weight from a pen. And so what we can do is actually look at the specific growth rate. And the really encouraging thing here is that blue mussel looks like it is actually, can be perfectly substituted for this fish meal. The food conversion rate, so they don't have to eat more of it, is also very, very good to fish meal. There are no changes in the microbiome, which is really encouraging. The only thing we did find out is actually it really depends how you provide this blue mussel. So you can either freeze dry it, which is a bit more expensive than crushing up the mussel with the shell. The fish really don't like the shell. I can understand that. So really, this just really shows you a collection of things we do in Magnify as a whole, and how we are using microbes and how we can actually start understanding how this all fits together in larger ecosystems or in respect to host. In this slide, I'm just illustrating all of the collaborations I have. And I think this is really the take-home message. Nobody knows everything, and you can't do everything. You have to find people who are far better at wet lab than I am to do these things. We can mine the data, but we need people to test. And I really would say that one of the key things throughout my scientific career has been the many fantastic collaborations. And I would just say, finally, thank you for listening. And leave it there, and I'll answer any questions later.
Thank you, Rob. Thank you so much. Thanks for building such great tools so that the community could move things forward and forward. And our last speaker before we move to the discussion and Q&A, which by the way you can contribute to, you can see there's an option to add your questions to any of the speakers, or you can also throw any oddballs you want of challenging questions that you want the perspective of the speakers, even if it's not directly related to something they mentioned. We're open to such challenges, so please feel free to do that. So before we move on, our last speaker is Professor Toby Erb, who is also the director of the Max Planck Institute at Marburg. He's really doing fantastic work as a leader in research on synthetic metabolic pathways that I'm sure you'll be amazed with very soon. But he initially planned to become a science journalist until he fell in love with the lab, where still he often finds himself, he was stuck with his experiments and had to go for long runs to help him. He and his team want to push the boundaries of what is experimentally possible, even though they are told that their ideas will never work out, and sometimes they do and sometimes they don't. Please tell us the most recent update, Toby.
Okay, so thanks again for this invitation. I'm really excited to speak about our research in which we want to use our understanding of natural CO2 fixation to create new ways, new to nature photosynthesis. Basically, rethink, redesign, and reconstruct new ways how light-driven carbon capture can actually happen inside of living cells. And really all our efforts focus currently on this small molecule here, CO2, which you all know, a very important greenhouse gas, and of which you also know that we have been started to concentrate it in the atmosphere in last years, decades, even centuries. So we human beings have been influencing the atmospheric CO2 pool quite a lot. And of course, you all know there's a dire need to find new ways to basically recapture this atmospheric CO2 and convert it, store it, or make something useful out of it. And that's a big vision, of course, for us to heal the planet, to come back to equilibrium. But of course, you also know the technological challenges that chemistry cannot simply just, you cannot simply run a chemical process to refix the CO2, which is so finely concentrated. And so we're still stuck in terms of technologies, at least chemical technologies, but you also know that there's actually a biological process that is operating at a large planetary scale. And this is, of course, photosynthesis. It's nothing else than light-driven carbon capture and conversion. And this whole thing operates at a level of 100 gigatons carbon per year, as Ron actually nicely calculated. So I'm telling you, there is a biological solution, and the solution is quite powerful, but still it's not enough to compensate what we humans do to the global carbon cycle. So the question, of course, is why is it not enough? Why is photosynthesis not able to compensate? And there's many thoughts and research done on this. Just want to focus on a very simple observation, maybe one of the main explanations, and that is that biological CO2 fixation is basically stuck in the way it fixes or captures and converts CO2. And it all comes down basically to the central enzyme, the central catalyst, if you want to say so, the engine that drives photosynthesis of carbon capture and conversion. And this is an enzyme that you all know called Rubisco, which introduces carbon dioxide to the Calvin cycle into plant metabolism. And what's interesting about this enzyme is, and I think Ron has also done all these calculations in the past, so this enzyme is quite a slow enzyme, at least slow compared to many other enzymes. It takes only 5 CO2 molecules per second compared to many other enzymes, which would be able to operate at 100,000 molecules per second. And interestingly, at the same time, the enzyme is also sloppy. It actually makes a lot of mistakes. And so it can, of course, accept CO2, but it can also accept oxygen. And in fact, an average Rubisco would have an error rate of approximately 20%, which means every 5th time, instead of fixing a CO2 molecule, Rubisco would fix an oxygen molecule. This would lead to a process called photorespiration. And this process, again, wastes energy, releases CO2, and limits, if you want to say so, photosynthesis by a factor of 20 to 30%. Now, on top of everything, a plant leaf is fully packed with this enzyme. And you can tell already from that, basically the Calvin cycle, biological CO2 fixation in the plant context is running at its limits. It's like a car you drive full speed, and it's pretty hard to improve that. I think many people have tried to improve Rubisco, have tried to improve the Calvin cycle, and we have not done that, but we have thought about a different way, trying an outside of the box approach and asking ourselves, instead of improving Rubisco, instead of improving the Calvin cycle, instead of improving existing photosynthesis, can we actually think of other ways, new to nature pathways, new to nature enzymes, newly discovered enzymes, new metabolic pathways we could think of that we could build and maybe realize in the context of a living organism. And this would be synthetic photosynthesis or photosynthesis 2.0, man or human made. All right. So of course you can ask, is this possible? Can we actually reinvent such a central process? And I can tell you, we take a lesson from the nature. And we can see that nature already has done this to some extent, because nature has evolved several different pathways beside the Calvin cycle, notably, of course, in microorganisms. Dianne is nodding her head. That's what I'm saying. Nature has been creative. Nature has found different solutions. And as a biologist, I'm amazed by these 7 other pathways that exist and run in different microorganisms. But as a synthetic biologist, I'm disappointed. I'm disappointed because nature missed out on many other opportunities. So you can look at those pathways or those enzymes, you can connect them a different way. And something that Ron and Aaron Bevan has also done in the past, thinking what else could actually exist and why did nature choose one or the other solution? And this is now real synthetic biology. You try to explore the landscape, the metabolic landscape, which nature has not touched or not explored yet. And it's different from metabolic engineering, where you try to improve something that exists. We try to build something new that doesn't exist in nature yet. All right, so how are you going to do this? Well, in principle, we have two phases. First of all, you think hard, what would be a perfect CO2 fixation cycle? What would a perfect photosynthetic pathway look like? You then move ahead and you try to find the individual parts, the enzymes, the proteins you would need to build the system, molecular components. You build the molecular system, the pathway that fixes CO2. And then you optimize it, and ideally you transplant it inside of a cellular system, which could be a natural cell, a natural microorganism, or it could be an artificial cell. I'll talk about both of it in a minute. And eventually, at some point, you're going to move it into a higher organism where actually scaling can take place. All right. But we start first with designing new to nature pathways. How do we do that? Well, in principle, we just sit really at a table. We have a white paper in front of us, a blackboard, and we just draw about hypothetical, theoretical, potential pathways. It's called paper biochemistry or metabolic retrosynthesis if you're a chemist by training. What you see are some of these ideas we've had, and I think Ron and Aaron had many other great ideas. The point is you can be super creative because you have so many opportunities. And so these are a couple of examples we've drawn. But of course, the question is which of these examples you draw is a good and efficient pathway. So which of those are really pathways that would make sense to build? Or if you're a PhD candidate, of course, which of these pathways gives you a nice thesis or a paper at the end of the day where it can be successful? Well, the point is we use very simple physical chemical parameters that we know from metabolic pathway design, that we know from natural physiology. We basically pay attention to kinetics, which means the speed of reactions. We want to use enzymes or pathways or reactions that are very, very fast and efficient. And one example is here. We don't use Rubisco, which is a very slow enzyme. We use another enzyme called ECR that can fix carbon dioxide that we discovered 10 years ago in a microorganism from the soil, which outcompetes Rubisco by a factor of 20, which means within the same time, you can actually fix 20 times more CO2. And naturally, a cycle that uses this enzyme would be able to cycle 20 times faster and fix 20 times more CO2. And second, we pay attention to thermodynamics. So the question is, how much energy do you have to spend if you want to fix one CO2 molecule? And again, you can make some predictions. You can talk about and think about potential ATP requiring steps, potential reduction steps, and you can have a matrix where you calculate all this energy that needs to go into the fixation of carbon dioxide to make a certain product. What you see here is that the catch cycle we had designed in the lab, the initial version of it, is actually 2 times more ATP efficient than the Calvin cycle, which means to fix 1 molecule of CO2, you need only half of the ATP that you would need in the Calvin cycle. And looking from that very simple parameters, faster and more ATP efficient, you would say this would be a very nice solution to be realized in the lab. Now comes the hard part. The hard part is actually how to build this system, because what we've done is just thought about a potential hypothetical pathway, and we need to find now enzymes that we can use to realize and put all this very complex CO2 fixation machinery together. And for that, we need the community. We need people who discover new enzymes. We need people to describe the diversity and the metabolic potential of enzymes. We need resources. We just heard from Ron that are able to browse to identify potential candidates to, at the end of the day, build the system in the lab. And I just highlight two resources. One is the Enzyme Function Initiative. The other is basically Brenda, a simple classical database. And of course, we just heard about Magnify, which is also a great resource for us. So searching for enzymes we can use to build the system. And we really don't care where the enzymes are from. If they are from alpha proteobacterium, methylobacterium, rhodobacter, if they're from gram-positive Clostridia, E. coli, archaea, human liver, really, we really just pick and pack the best enzyme in our mind that we could use to build this system. And all together, to build the first version of the catch cycle, we combined 15 different enzymes from 6 different organisms and even one engineered enzyme to build the first version of the cycle. And we could really show that if you bring all these 15 enzymes together, you throw in energy, ATP and ADPH, and CO2, the system starts to turn and it starts to fix CO2, which is great because it tells you you can really rethink and rebuild a complete new CO2 fixation cycle from first principles. However, what we also learned is that our design was heavily flawed because the CO2 fixation efficiency was super, super, super low. And even though we thought so hard about the individual enzymes, we did not manage to build a biological system that was operating at high efficiency. And why is that the case? Well, the challenge is that biological engineering is not physical engineering. The parts are not perfect. The parts are not catalyzing it without doing any other stuff, and the parts co-evolve with each other. What I'm telling you is that if you bring in things from very different biological backgrounds, you need to spend a lot of time to optimize the system, or in other words, you need to mimic evolution to make the system more robust and more efficient. And this is what we did in the lab over different rounds, building a version 1, 2, 3, 4, 5, and just implementing things we already know, shaping, for instance, the specificity of enzymes, redesigning certain parts of the pathway, or introducing enzymes that remove toxic products to make the system more robust and improve it by a factor of 20, but just implementing all these different principles that we know that evolution is playing on. So we built a system now with 17 different enzymes from 9 different organisms, 3 re-engineered enzymes, and the system in vitro is already at the level where you would be able to measure if you just measure the Calvin cycle and you break, let's say, plant leaf and try to measure CO2 fixation. That's very impressive, but of course it's not the end, right? In real evolution, you could go even further. The problem is, how do you do this in such a complex setup? So how can you systematically explore the optimum of your system? And this is, I think, where, again, bioinformatics, lab automation, and machine learning comes into play. So what we did in the last year is basically setting up many different variants of the cycle using an acoustic dispenser, miniaturizing the catch cycle in vitro in very small 384-well plates, and running hundreds to thousands different variants, analyzing which of these different variants shows the best results, feeding it back into a machine learning algorithm, and letting predict the next set of experiments. And what you see here is, over the 8 to 10 days, you actually improve the system by a factor of 10, just using now, let's say, machine learning to mimic evolution in an in vitro setup. And now I think we are really at a stage where we can really think about implementing a system as a robust system inside of a natural or synthetic cell. Well, this brings me now to the comparison, what is happening in natural chloroplast and what's happening, let's say, in an artificial chloroplast. So how far can we actually get in building a light-driven, light-powered in vitro system? LD, you need 2 things. You need to power the system with ATP and NADPH through light, and you want to confine the reaction inside of a compartment like in a real chloroplast. So the first part, can you take a synthetic cycle and hook it up to the photosynthetic machinery of spinach, for instance? So can we use the synthetic system and couple it to the natural system? Actually, it's not possible because, again, there is no co-evolution, and you have to think hard and identify the weak spots and those enzymes that make problems. You switch parts again, 2 parts in this case, and suddenly the light-driven machinery of spinach chloroplast can fix carbon dioxide in combination with our synthetic catch cycle. We can further encapsulate the whole things with a technology called microfluidics, and we create really inside of very small cell-sized droplets a system where we have the thylakoid, the chloroplast, membranes, as well as our synthetic cycle. We can shine light on these droplets. We can power them up, and you see here's NADPH, that's the energy source. And then the NADPH can be used by the catch cycle to fix carbon dioxide. And we actually can show that this works at least for 2 hours. All right, this is the idea of an artificial cell, an artificial chloroplast, a human-made, let's say, biological-like structure at the interface of chemistry, biology, material sciences. But of course, it would be even greater if you could take now this new to nature pathway and put it back inside of a living cell. And so, or another comparison would be, let's take this pathway, this new software you've just designed, and try to run it on existing hardware. Let's say an E. coli cell first, and then maybe later on in a plant. So how are we going to do this? Well, this is work that has also been pioneered by Ron in the past. You actually make the cells addicted to your pathways, right? So you, let's say for the catch cycle, you isolate one metabolite that's essential for the cell. In this case, succinyl-CoA, a citric acid cycle intermediate. You create all pathways leading to succinyl-CoA, and the cell is really dependent on this compound. And you bring back in now the catch cycle or parts of the catch cycle. And only if the catch cycle is functioning, it would allow the cell to make succinyl-CoA, and the cell could start to grow again. And this, in fact, what we've been doing the last 3 years. And lo and behold, we could really show that in the meanwhile, if you put in the catch cycle in E. coli and we make it addicted on it, it really starts to grow, which is the first step of bringing this new to nature metabolic pathway network into the context of a living cell, and hopefully harnessing and leveraging evolution to further improve it in the future. All right. One thing I want to mention, of course, is even though we have made a lot of progress thanks to many great scientists in our team, I think one thing is for sure the small droplets or the E. coli cells growing a little bit in a flask in the lab won't save the world from global warming. But we have learned what are the building principles of metabolic networks. We've learned what does it take us to rebuild and redesign new metabolic pathways. Can we mimic evolution, and can we transplant it inside of living cells? And I think we've gotten a lot of insight we can use now to think about real world application in the future. And those might be down the road 20 years from now, and you might be the people implementing it and harnessing these principles. But I think we're on a very good track, at least in this respect, looking at this design, build, and implementation phase. I want to put at the end of my talk, you have 2 more minutes, just our research, again, in the context of the overall session of the great talks by Dianne and of course also Rob. And I want to say, we have been focusing very much on the carbon cycle, on new ways how to fix, capture, convert carbon dioxide. But of course, there's many other elemental cycles that are heavily influenced by us human beings. You might know nitrogen cycle, right? You might know that phosphorus is an ending resource. We are hitting planetary boundaries and we're influencing all these cycles. At the same time, those cycles are, of course, influenced heavily and shaped heavily by microorganisms, which are the key players. And I think we should be just humble enough to learn from the experts, how the experts keep those cycles sustainable. And this might guide us to new ways to heal the planet. And I think I've identified 3 challenges you might drive on in our discussion. I think one challenge would be, first of all, put forward by Dianne. One thing we need to do is we need to bridge the scales in microbiology. On the one hand, on the right side, this is CO2 fixation of Synechocysts chlorococcus, marine algae, right, at the planetary scale, 10 to the 7. On the left side is research from our lab where we have identified how an enzyme can bind, capture, and fix in carbon dioxide at the active side, 10 to the minus 10, 17 orders of magnitude of difference, but we still cannot connect the left side with the right side. And I think you might be those people that can come to unifying framework and can really come from atomic structures to global processes. Okay, we need to understand how things are connected from the smallest to the largest scale. Second challenge, of course, put forward by Rob, also by Dianne. So what are all these freaking enzymes doing? We're discovering all the billions of genes and billions of proteins we can now predict. So what is their function? How flexible are they? What are they doing there? And I think this is one of the biggest challenges for me as a biologist. At the same time, one of the biggest joys for me as a synthetic biologist to harness all this power of evolution, hopefully. But it's also a challenge for us as a community. How do we evaluate people who are really sitting there and trying to find a single function of a single gene? Doesn't give you sometimes a Nature paper, but it's really important to do. How do we incentivize these efforts really and pay back those hard-working biochemical people in the lab? And last but not least, of course, thinking of applying all these new enzymes in new contexts, how flexible biological systems? Can we rewrite the code of life? Can we put in new functionalities? Can we basically reprogram the operational system of cells? Or do we need to develop minimal cells like I showed in the beginning to come to new functionalities? At the end of the day, I think, just to come back to microbiology, I think new solutions to environmental challenges of today can only be basically solved with the help of microorganisms. And I think that is my take-home message. And I hand over to the panel, and I'm happy to take, of course, questions. Thank you so much.
Thank you, Toby. Thank you very much. So I think it's a good opportunity to say, on the one hand, thanks for all the 3 speakers for great talks, but also to sympathize with the listeners. Maybe it's a good time to take a good breath, maybe to have a stretch. There's a good chance, at least that happens to me all the time, that even when the speakers do their best to explain, I feel saturated and overwhelmed. But at the same time, we want you to stay with us for a few more minutes because we want to have both a discussion what we saw and to see if there's questions coming up. So if you feel like, it's overwhelming, it's like what most people feel. And there's a very known thing that happens to people in our profession. It's called the imposter syndrome, where people feel that actually all the others, all the hundreds of people listening to this really understand everything and follow everything, all the slides. And only they are kind of slow and dumb because they couldn't follow what was happening here. So I have a surprise for you. It's already has been researched and it's been found that really the vast majority of people are feeling that the same feeling that you're feeling right now, that this is too much for you. The other people are smarter than you are. It's very, very common. If you don't believe me, Google it under the imposter syndrome. Okay, and in that spirit, I think we want to challenge the panelists with either your questions or some broad view things for discussion. And we might also have some extra websites and resources that I think Janine is highlighting. And if you want to follow some of those websites, they're the most, the best things that our speakers thought that would be of most value. So I'd like to, okay, so everybody took the breath at the time for a stretch. You can do it, in any way you want because we don't really see you. So that really gives you even an advantage over all of us speakers. And I want to start with trying to think about, for each of the speakers in their field, something about the best case scenario as a community. So trying to think, be realistic, but still optimistic about, which environmental challenges you think we could tackle. If you have a quantitative bent to it, I think that helps because usually things are on a quantitative spectrum. They're not just black and white. And if you could say something about where does life scientists, which is much of our audience, where do they have leverage and to which type of questions? So you can either jump in or we'll go with the order of the speakers of Dianne and then Rob and then Toby. But if somebody wants to jump in, you can go ahead. I think you can all unmute yourself.
Okay. Dianne, you want to go ahead or?
Sure, I'll just pick up where you left off, Toby, because I really loved your final sets of slides, and I thought it was wonderful how you highlighted that in addition to the carbon cycle, the nitrogen cycle and phosphorus cycle are very important, and of course there are other cycles too. But maybe that's a good place to begin. So I do think that it is important for us as a community to identify where the issues are that are actually tractable based upon the type of experiments and knowledge that we as life scientists can get. And I like the phosphorus and the nitrogen cycle and the carbon cycle very much because I think all fit that bill. So as you pointed out, we're running out of phosphorus. When that will actually happen is not entirely clear. Some estimates I've seen on the order of a century. Certainly, it behooves us to be able to use phosphorus much more efficiently and sustainably. And I gave one example from my talk of how we might take a hint from microbial strategies that clearly are ones that potentially have something to offer, but that was just one example. There are many other examples where fungi, this is a reason I provided the link to Toby Keir's website, fungi are other microbes that don't get nearly enough airtime that are absolutely vital to the health of plants through intimate symbiotic associations where they help deliver not only phosphorus, but water. Thinking about water delivery, especially as we're going forward now with global change, there are going to be certain parts of the world that are going to become much more arid. Thinking about how we can tap into microbial communities to actually capture water more efficiently. I think these are topical areas that are actually very important. Numerically, Ron, I don't have a number off the top of my head that I can give you. I put that to you as a challenge. Maybe you can say more on that topical aspect, but I think actually I don't know if it's understood. I don't know if we know well enough, and I think that's something that we as a community need to think through a lot harder, which are what are the problems, what do we think are the potential solutions, and then do the calculations to ask if we improved phosphorus recycling, for example, in a soil by X amount, what would that translate to in terms of ability to conserve phosphorus resource. I mean, you could do that kind of analysis on many topics. I'll stop and give the baton to someone else.
Right, maybe I jump in. Or Rob, I don't want to skip you. So my number in my head is 8 tons per CO2. I think we should all get used to think how much CO2 we are emitting, as much as we sometimes start to count calories in these days, right? So we need to get a healthy understanding what each individual is contributing to CO2 emissions. This is the first step to get to become more quantitative and also scientific in the ways in making decisions, right? For instance, that we meet now online is a very good decision because I think we saved a lot of CO2. So I also think about other numbers, which are how much energy comes to this planet. And Dianne, you know this better than I do. Ron, you know it even better than Dianne probably. So it's 100 watts per square meters, at least in Germany, right? And we're just able to capture 1% of it. So in principle, it's enough energy there, right? But the question is, how can we become more efficient in harnessing this energy and using the energy on this planet to influence, in my case, of course, the carbon cycle. So can we, for instance, combine chemistry, photovoltaics, together with a biological system? Can we create this biohybrid system? Some people, especially also Ron, is working on. And can we leverage the power of biology in combination with great material sciences? And again, I think this is a vast untapped potential for us as life scientists to move into this area. And I'm just fascinated by the possibilities in there. And I think we should always be open to learn from nature, but also think how we can improve and or implement these natural principles in, let's say, more technological setup. And to give you one last example why I think it's important. So we all know how birds fly. We're all amazed by the flight of birds, and we've studied it for centuries. At the end, we build planes. They don't look like birds, but they mimic those principles that we've learned from the natural system. And maybe the CO2 fixation principle is not, let's say, photosynthesis, but the principle of CO2 fixation in a more technological setup. And I think we should not stop thinking about how to use those principles we learned from studying natural systems and to create new solutions. And this is, I think, what I would find most inspiring as a biologist these days. Okay, Rob.
Yeah, I mean, those two sort of comments are tough to follow. I would say something maybe more on sort of following on in terms of, why do we care about microbes so much? And I think the reason being is that microbes have basically exploited every single niche on this planet, and they are highly adaptable, and their ability to change due to their relatively short generation time is something that has made them so successful as a set of organisms. But as Toby mentioned, in terms of things like plastic, it's a really new substrate for these microbes to see. So there are some, what we're currently, the area where we're looking at are finding very sort of generalist enzymes. And then we have to then use the techniques, high throughput functional screening with mutagenesis to then kickstart that to try and get them to adapt. So you're accelerating that evolutionary process. We shouldn't be scared of doing that, but we also need to be just mindful of how we present that to a wider audience about what we're doing and what this represents. Everyone was probably familiar with the genetically modified crops and how society misunderstood to a certain extent what they were bringing, and that you can get a really negative pushback if we don't engage appropriately. So I think there is a little bit of, we've got huge potential there. I think the adaptability of microbes and the ability for us to genetically manipulate them is fantastic, but we just need to do this in a considered way. And I think there is also a notion of, we have to be mindful of the digital sequence and making sure that Nagoya is reflected. So this is the protocol to make sure that there is benefit sharing across the world. So companies going, companies going in, mining these data sets, they should not be allowed to just make a massive profit because that sequence has come from an indigenous country where maybe they haven't got the capabilities to exploit it.
Okay, I think I got much more than I asked for. So we've got perspectives both on where the life scientists, but also touching on some of the real challenges that we're facing as science wants to make an impact and acceptance, etc. I'd like to also have people touching on things they've seen in their field that is inspiring to them. It could be some study, it could be some website, it could be an initiative that they want to highlight. So now is your opportunity to do that.
I'll go first, sure. Someone needs to talk. I have given a couple of websites, both in my talk and have been provided. I encourage you all to look at them. These involve quantitation of human impacts on the biosphere, and also a wonderful website looking at the Society for the Protection of Underground Networks, talking about the importance of soil fungi. But as an example of research, to just get back to the point that the microbial world is really quite extraordinary as it is, and if we understand how things are limited, we can control them better. I'll point to research that was done in, I believe, 1993 in an experiment designed to try to understand why there were regions in the equatorial Pacific that were so-called high nutrient but low chlorophyll, where there was high nitrogen, high phosphorus, and yet there weren't very many phytoplankton growing. And the answer turned out to be that there was a limitation for iron. And so an experiment was done where a boat went over, like 60-something square kilometers and put experimentally into the oceans a certain amount of iron chloride. And sure enough, there was a resulting bloom in phytoplankton. Now, one can debate whether or not this is a good idea. One would need to know the unintended effects of something like this. But what I think it demonstrates, and the reason I bring it up, is that as we better understand what limits microbial processes that we care about, we can think about how to stimulate them. And so there's an example of something occurring on a large scale due to the insight that came from knowing that trace metals can limit microbial growth.
I'll pitch in with a different site. So I'll pitch in with the Tara Oceans project. So I passed the URL to Janine, and so I mean, I think this is a really good example, or the best example of where a global initiative of open data and really well-structured data has really sort of given us that ground zero of starting to understand the health of the world oceans. And I know there was a previous session on marine microbiomes, and that was probably covered to some extent there. So I would say that that's a really good example. If you want to start thinking about a multi-omics, multi-faceted project that has then continued and grown, then I would really recommend going and looking there. And they are tackling different issues like plastic pollution now in the world's oceans. And I want to make sure Toby's got enough time.
I mean, the great points you all mentioned, I can fully subscribe to your statements. I want to give a different angle to what both of you have said, and maybe also emphasize technological opportunities. When I did my PhD, I mean, we had to clone genes by hand, but how hard it has been. Now we just order genes, right? So also the high throughput capabilities, the microscope you can use, the metagenomic, genomics, proteomics, machine learning, big data and analytic capabilities we have, speed up the discovery and also design of our experiments. And I think being more quantitative about is very important. Being also a bit more synthetic is very important because it allows you to test hypotheses. And I ask myself also, how does the future of lab look like in 10 or 20 years for us, or for you as trainees right now, right? So we can leverage the power of living systems by manipulating them really hard. And Ron has done great examples of showcasing this and learning again back what happens if you challenge those systems, which tells about the flexibility and how fast things can adapt. And this is something which is pretty hard to predict these days when we change, let's say, the global carbon cycle so fast. I mean, there is a natural limit how fast organisms can adapt to that. Microbes might be the best in it. Rob pointed it out. They've been around for 3.5 billion years, but we also know this is probably not us human beings being adapting so fast. So I'm just amazed by those works that also try to do experimental evolution. And what came out of Ron's lab was really fascinating for me. And I think this is probably maybe a good ending and handing it back over to Ron.
So I see also in the questions that we're getting a lot of emphasis on evolution and how can we consider doing something that would be, on the one hand, leveraging from evolution And in that respect, there's the challenge that many of the microbes that we might want to leverage are actually, there's a question of whether we could culture them in the lab or not. So maybe on those challenges, anybody wants to reflect. And at the same time, maybe another related question is about how could we, given the 3 billion or 4 billion years head start, how can we think that we might be doing things differently, better? What is really happening here? So maybe touching on that question from the audience.
I would love to jump in on that because I think that oftentimes there's a statistic floated around that only 1% of microbes in nature are able to be cultured. And I want to demystify this because I think that that's a number that has been overplayed and may not be accurate, even when it was first put out. And it certainly isn't accurate now because we're living in an era where there has been tremendous acceleration in our ability to understand the environmental chemistry in which interesting microbes reside. We have a much better appreciation for their interactions with each other. Technologies have advanced so that we're able to cultivate organisms at the single cell level now, allowing slower growing bacteria or other types of microbes to be separated and brought into isolation. In addition, there have been unbelievable advances in genetic technology so that now it's extremely easy to be able to manipulate just about any organism. So these things together provide just unbelievable opportunities to go into nature and to enrich and select for catalytic abilities in the microbial world that are of interest, which is sort of the first step towards being able to later do synthetic biology and engineer and adapt them, as Toby explained. So I think it's an exciting time to be a young person in this field. And I'll end there and see what others say.
Maybe I should demystify our picture of evolution as being the most creative force on this planet. I fully agree, right? But you should not forget how evolution works. It always builds on existing parts, on existing subparts and existing systems, right? And so in the beginning, it might be really rapidly scanning, let's say, the space until homing in on one certain solution. Then it will evolve and improve this certain solution, finding a local optimum, but not maybe the global optimum. It's us as human beings being very progressive as teenagers and becoming very conservative at the end. So, and being able to replay the whole system and setting it to zero and seeing how fast it could evolve, this is fascinating to me. So what I want to say is there's this strong historical dependency. If you evolve a new metabolite in the context of a metabolic network, you want to make sure that it doesn't disturb 3,000 other enzymes in the context of E. coli cells or the genetic network. So there's a lot of constraints. Evolvable a system is once it has reached a state, like after 3.5 billion years of evolution. There's Windows 11, Windows 13, Windows 14, but maybe it's better to have an iOS system being built or Linux at the end of the day to restart new possibilities. And I think this is what we also should appreciate as biologists, that evolution, yes, is creative, but it's also maybe homing in and evolving in one direction. It's pretty hard to turn it back around. It's a one-way road to some extent due to dependencies that exist. And David Baker has shown that. David Baker has shown there's many more protein folds that could exist in theory, but were never used by biology. It always builds on, let's say, 10 or 20 different folds and reshuffles those folds, but doesn't move into this other space for what reason ever. So don't overestimate the power of evolution. That's my 2 cents.
So I'm going to come back to Dianne and just on her point. I agree that the way that things have yet. So I always refer to it as yet to be cultured, not to say they're unculturable. That's always my way. And what I would say is actually that we've already started having some success in looking at some of these poorly represented clades and actually using just the metagenomic information to actually guide the culturing techniques to actually you work out what is this microbe deficient in terms of pathways to then say it should have this. I think the notion of actually going across, going away from monocultures and having simple communities is going to be the next big wave in culturomics. But I would just refer back to the my protein database in terms of when we talk about sort of 2.4 billion sequences. It depends if your glass is half full or half empty. I'm going to be the pessimistic person today. So only half of those we can actually put any form of functional annotation on, which means the other half we have no idea. They're just a sequence. And I think that to me is the challenge. And Toby, you mentioned it as well, in the fact that, what are all of these doing? Now, we are beginning to understand a little bit about what these are doing. And I think there are a lot of those things that don't get labels, of just some of that is bias about how we label things. I think there are still some really interesting pockets out there of things that are either very different to what we know or are completely new. And I think that is both a challenge and an exciting area that I would want to go into if I was a bit younger.
Fantastic. I would suggest that you maybe consider it, no matter what's your age, to take on another challenge. I think we will soon be moving into the opportunity that we have for the audience to meet the speakers. So we'll take just a few more minutes, and then we'll move into the next stage, in which you can really get to know people in a different mode rather than just this one. There was a question that was geared to Toby about whether we could expect nature and evolution to have found other pathways for CO2 fixation that might be found in the near future. Do you want to mention something about that?
Sure. So I want to start with that. Let's say genomic and metagenomic evolution has allowed us to look and find bugs that can grow with CO2, a source of carbon, but apparently don't use the Calvin cycle, which has led to the discovery of 3 or 4 new CO2 fixing pathways. I also want to mention that, let's say 2-3 years ago, a couple of years ago, we also had synthetic biologists like Aaron Bar-Evan thinking about pathways like the reductive glycine cleavage pathway, which was designed first on the paper and then found later as being actually existing in nature. So it tells you already that some of the things we think of, maybe the catch cycle, could actually exist also in natural systems. But knowing how these systems look like, we can now harness and go through metagenomes and look which bug might have all the key enzymes required. And that's of course exciting for us too, to see if there's even more pathways out there. It's hard to find them, but we can find them. And I think we've seen over the last 10 years, 3, 4, 5 new pathways being discovered. The question of how big is the niche you're living in, right? I think that is of course defining also the success of the discovery. Yeah.
So thank you for the Siva Sanka from the audience for bringing up this question. And another question that was raised was in relation, maybe it's relevant for all 3 speakers, about the fusing of enzymes. So Orakov Askalbek is asking about whether fusing of enzymes could improve pathways and efficiency. And I think that might relate to each of the speakers today.
I can go. So fusing of enzymes to complete a pathway presents challenges. So usually within a pathway, there's one rate limiting step. And so if you're constantly fusing things together, you've then got a pair and you're carrying around something extra that you don't necessarily need to do. Often making a fusion also limits the dynamics of your substrates flapping around. It can also change things like that. So I think that, there are different techniques that I hear about in terms of actually having really smart systems of actually how you can cascade substrates from one as they're getting processed. And I'm not the person to answer that. I think I'll hand the sort of more wet lab answers to my esteemed colleagues.
I'll just take this as an opportunity to point out that classical genetics and biochemistry really never go out of style, and that if we want to fundamentally understand a catalytic process, we need to do the hard work to figure it out piece by piece, because the answer is going to vary depending upon the pathway that you're talking about. And so it's very difficult to generalize, and it's important to be precise. But this is where my optimism returns, is that I think that nowadays there's an opportunity to do this type of mechanistic research with a glorious diversity of microbes if we're selecting for phenotypes that we care about. Where we have a phenotype, there is now a path to figuring out how it works. It requires a commitment, but it's an incredibly exciting opportunity for fundamental discovery.
I would love to mention and to thank back the person from the audience. The name is Vesta Patrasionate, who was mentioning the big thank you for the organizers. So I'm not one of the organizers, and therefore I could thank them also on everybody's behalf. So it's a really collaboration between the HHMI, the Howard Hughes Medical Institute, and the EMBL, the European Microbiology Laboratory, which I think, if you're interested, everything that you saw now, if you want to share with everybody, that's been recorded and will be available on the website. But also, this is a part of a series of lectures with institutions, leading scientists from around the world on varied subjects. So you could share also that with many other people that I think might find interest in that. And that's one of the beautiful things in the world today, how it's so, not only is it not only cheap, it's all free, it's available, all of that knowledge, insights, etc., to everyone. So thanks to the organizers, and we'll now take the next few minutes to have the opportunity for people to interact directly with the speakers. So we'll be led into the Meet the Speakers session. Thanks everybody, have a wonderful day, and choose good problems and get them done.
Wonderful. Thank you guys so much. Thank you to you, Ron, and of course to Dianne, Rob, and Toby for a really fascinating session today. Thanks to our whole audience for joining us. As always, I do want to mention that all of the websites and resources mentioned by the speakers in their talks are noted in the document that I shared in the chat box, and they'll also be available on the Life Science Across the Globe website, along with the recordings of today's session. If you would be so kind, we would love if you would take a moment to complete the survey that I linked in the chat box. And also don't forget to join our next Life Science Across the Globe event, which will take place on June 1st at the same time, 9 a.m. Eastern, focused on air quality and infectious disease, hosted by the Institute of Infectious Disease and Molecular Medicine at the University of Cape Town in South Africa. So we're really looking forward to that.